Thank you, Marcy, for the introduction. And I'm glad that we are back for the first choosing tag of the semester. And as Marcy mentioned, I'm a recent graduate from Federal Lab. And if you went to my theater defense, you might find some of the things pretty familiar, but we're just talking about how choosing tag is like more like you can Q&A throughout, and you can talk about developing tools that are still actively being worked on. So I would like to hear all of your questions throughout the presentation. So my talk today is going to be on a flexible modeling framework that uses a zero-inflated negative binomial mixture model. So before we go into the tools part of it, I want to introduce this whole thing with the data that led us to this tool. So we have a collaborator in the Department of Biological Chemistry that worked on CRISPR-Cas in Nigeria. And CRISPR-Cas is in bacteria, giving it kind of like adaptive immunity. It can be kind of like set into 3 different stages. The first one is adaptation, where the foreign DNA is first recognized, excised, and then integrated onto the native CRISPR array and become new spacers. And now it is part of the immunological memory that the bacteria can use to defend from future invading DNAs. And the second stage is CRISPR RNA biogenesis. And the third stage is probably a lot more people are familiar with, is the target interference stage, where the foreign DNA is recognized and then cleaved. So as we can see here, that new spacer is really a very basic part of this process, because that's where the memory comes from and where this immunity is going to work on. And if we really tune up the activity in the of the CRISPR-Cas system in the bacteria, it is possible that it take up self-DNA as new spacers, because that's probably the most abundant DNA lying around is inside the bacteria itself genome. So with that, we are more interested in the adaptation process, and we want to know how our DNA donors selected for the immunological memory. What criteria makes the DNA more susceptible to become to be recognized by the CRISPR-Cas system? So what we're interested in is basically the newly integrated new spacer, as in the yellow box. What are the surrounded genomic contacts, and what are the sequence features that comes along with it? So one of the first things we think that we'll check is that across the genome of the self-DNA, is there a differences of where new spacer is coming from? Because if there's no difference, it's most likely the selection is random. And we look across the genome coordinates and plot how many spacers are there at each genome coordinate that we found. So the first thing we notice is that there is a range of different level of preferences of different genomic sites being uptaken as new spacers. And some of it, some of the hotspots, like the very high peaks, we can associate with genomic features that we already know. For example, on the very left there is the origin of replication, and in the middle, that is roughly where the terminus is. And the third one is where the native CRISPR array is. So we think that it is likely it's related to single-stranded DNA availability to for the CRISPR-Cas system to uptake a strand of DNA into as new spacers. So that is the data we see, but we want to know with the sparse annotation and everything, and we want to know, do the genomic sequence and the chromosomal features play a role in the spacer acquisition preferences? So that's the data part. And that's what led us eventually into dealing with this question on a more statistical point of view. So the first thing we did is that we curated a list of genomic features, including that if a genomic site is in the gene or not, its local GC scale is 80 content, what's the DNA shape predicted around it, and what is its distance to some important genomic features such as origin of replication, etc. And we calculate the Spearman correlation of such features with the spacer tracks that we just saw. And we want to know whether or not this correlation is actually meaningful. What we did is that we did a permutation test. We shuffle the blocks of genomic features and recalculate the Spearman correlation over and over again that establish a baseline of what we expect if there's not really any correlation. So we can see that in this example of AT content, there's a small correlation, but it's robustly away from what we expect as the baseline. Similarly, we do that for the rest of the feature, and many of them, as for example, the distance to the origin has like a modest correlation that is robust. But in the case of like the distance to the origin replication, that is intuitively similar to what we see visually in the track, where there's a hotspot at the terminus. So these are the features we're currently working with, and we cannot just do Spearman correlation over and over again with all the features we find. So our question becomes, how can we systematically identify possible factors that affect this CRISPR spacer acquisition preference? So this is where the regression model comes in. Basically, what we want to establish is that given an independent variable, in our case, that will be a certain genomic feature, we want to establish its relationship with the outcome. In our case, is the spacer count. So there are a lot of like benefits of using a regression type of approach instead of, again, doing correlation over and over again for individual features. For example, we can actually investigate the interaction between different terms, different independent variables. It will give us better handling of replicate data and also discrete variables. And a little bit along down the line, we can compare different models to really understand if a variable is really contributing to the model and contributing information to the spacer count. So that is when the regression model comes in. And to do regression model, the first thing we want to check is that what is the distribution of the spacer count? What does it look like statistically? So what we did is that we take the spacer count and we count the number of genomic sites given a certain spacer count within different intervals that's shown on the x-axis. So here is the bar chart showing the spacer counts in different bins. And very strikingly, we notice there's an excess of 0 of spacer count. It is possibly coming from either a lot of genomic sites are not taken up as new spacer at all, or they are not detected. There are a lot of reasons for that. But given the big genome, this is not surprising that we end up with a lot of like sites with 0 spacer counts. But this clues us in that a zero-inflated model may be a good place to start. So we wanted to start with a zero-inflated negative binomial model. And to use a mock example, so let's say you have a pile of metal beads and you have a plastic cup, and you are going to scoop the beads using the cup and then count how many beads you get in each scoop. So in this process, you will end up getting a Poisson distribution. And now let's say those metal beads are magnetic and they tend to clump with each other. Now you have like over dispersion coming in. And the same process will give you counts that follow a negative binomial distribution. But for zero-inflated negative binomial distribution, you need to think about it like you also have a non-functional cup that has its bottom cut off. So every time you try to scoop beads, you just end up getting 0 counts. And you repeat the same process. Now you have 20% chance getting the non-functional cup and 80% chance getting the functional cup. What we will get is a zero-inflated negative binomial distribution where there is not only just a count component, but also a 0 component as color-coded here. So this is basically the model we want to start with, because given the excess of 0, and given that we are dealing with count data with a considerable level of dispersion. So what we did is that we fit a zero-inflated negative binomial model and compare the observed spacer counts with the predicted counts. So we see that in the predicted count, which is the purple histogram, there is a dramatic rightward shift compared to the observed count. We think it's probably coming from the negative binomial component of it, trying to account for the very high count we see in the data and end up being very dispersed in this process. So we look into our data and we concluded that this model is probably not sufficient to handle the complexity we see in the data if it has to use over dispersion to kind of compensate for it. So how can we make our model more reasonably complex, but also useful and statistically make sense? And this is where the zero-inflated negative mixture model comes in. So going back to our mock example here. So now, besides a non-functional cup and a functional cup, you get a third option that you can also randomly grab a bigger cup. So now you repeat the same process of scooping bees and then counting them. But now you have 20% chance getting the non-functional cup, which is going to be the zero inflation, as you already seen in the spoiler. And the small cup for 50% and 30% chance of getting the bigger cup. Then what you get is something follow a negative binomial mixture model. There is not only a zero component, and there are also two negative binomial components that make up the counts. It's also worth noting that the negative binomial component themselves is possible to generate zero counts. So not all zero counts come from the zero inflation part of it. And this is what we landed on a zero-inflated negative binomial mixture model. And we built this model and implemented it with a variable number of negative binomial components available. And that is where the tools and text part comes in. So we got there. And one thing we tested on is an RNA-seq data of an anaerobic biofilm culture. So what I want to see here is some of the results we see using this model with a considerable higher level of complexity that it can deal with. So we reanalyze this RNA-seq data. We quantify the gene expression with risk per gene here. We plot the histogram of this gene expression count, and we end up with something that looks like this. And by this time of the presentation, I probably talk about zero way too often, so you might notice that there is an excess of zero at the very far left of this graph. So wouldn't it be nice if the zero inflation got taken care of? And the distribution roughly follows a negative binomial distribution, but again, given the inflated zero, if we use existing tools to fit either a negative binomial distribution or a zero inflated negative binomial distribution, which has only one count component, we'll get the dashed line for the negative binomial distribution and the solid line for the zero inflation. So one thing we notice is that if you look at where the peak is in this fit, it is leftward shifted and is a little bit lower than what we observed. We think it's likely that it's trying to account for the zeros by shifting the negative binomial distribution and end up with this fit. So this is not ideal. And after we now have an implemented zero inflated negative binomial mixture model, we fit this to the data with from 1 to 4 different number of negative binomial components in the model. So here is the fit that we see. And in the orange line, we're with one negative binomial component, it still looks better than the existing tools. That's because in our tool, we implement a noise term that can handle outliers a little bit better than what is currently available. So that's also why our zero inflated one negative binomial component fit looks different than the existing tool. And the noise handling component so far has been proven pretty useful in our testings. So what we see here is that if we look at the peak, it fits a lot better than the other ones. But also more importantly, around where the zero is, our model is more successful in accounting and predicting the inflation in the zero counts. So that is the distribution part of it. So we have a statistical model, we have some data with count. We can fit this distribution of the gene expression data pretty well with our model. So now let's introduce some independent variables and see how regression works. So we created some gene level features for this data and try to do regressions on this set of data to see if we can establish a relationship between the independent variables that are the gene features and the gene expression counts. So in this process, there are two sets of parameters we can fit to. And then that means that for an independent variable, there are two ways it can affect a specific count. One is through the means of negative binomial components. So if we go back to the mock example we have, that will be the size of the functional cups. Basically, for the count components, what is the mean of the negative binomial distribution flaws? And in this mock example, for example, on the right, you see the distribution. The blue has a mean of 5, and the green one has a 15. And you can see they distribute quite differently. And if you fit an independent variable to the mean of these components, it can affect the eventual outcome through the means. So that is one way that an independent variable can play a role. So what we did is that given the gene level features we curated, just for examples, we have the length of the gene, the AT content, was the gene's distance to the origin of replication, and also whether or not a gene is annotated with anaerobic respiration functions. And we look at the values of coefficients that come with this regression model, which means that if we have a positive coefficient, the increase in the independent variable, in our case gene feature, would have like a positive effect on the eventual mean of the count. And I see that Marcy has a question.
Yes, there's a question online.
Yeah.
From Ned, who said, what each component in the model represents in the data acquisition process?
So that is actually a really good question. So one of the reasons we end up first finding this RNA-seq of biofilm anaerobic culture data is that this kind of complexity works really well with either clusters of genes that are co-regulated, but in our case, we are looking at possibly subpopulation of cells that has different gene expression patterns. And that is a possibility what different components can mean. And it varies from data to data. And in our case, for spacer acquisition, where we eventually want to land it on, our other study shows that there are more than one possible kind of like mechanism spacer can be uptaken. If we see there is the complexity more than just one count component, it is possible that those counts are following kind of a different mechanism with a different path that end up being uptaken as a spacer that follow a different set of molecular rules. So I wonder if that answered the questions online.
We will assume it does. If that doesn't, please
All right.
add something more in there.
A question?
Yeah, I was wondering,
Yeah.
why, did you, test, zero-inflated Poisson models?
Yeah.
As well, or what was the reasoning behind choosing negative binomial over Poisson?
So at the beginning of this project, where we started the development of the actual tools, we tried Poisson negative binomial distribution, both of them, and zero-inflated Poisson and zero-inflated negative binomial distribution. And we go with the statistic route of like checking the residue, checking the goodness of fit. And it has been pretty consistent between Poisson and negative binomial distribution. Negative binomial works better. I think it's coming from the dispersed nature of our counts, because we have a lot of small counts, but we can also end up with counts that on the scale of 5,000, for example. So with that, it is probably why a negative binomial distribution compared to Poisson handles it better. And we see similar result with the zero-inflated ones. That answer the question?
Yes.
All right, thank you. I appreciate the question. So definitely let me know if you have any thoughts. So in the regression model, the coefficients here is that if we have a positive coefficient as your independent variable increase, the outcome also increase. And the reverse for the negative ones, your independent variable increase, your outcome decrease. And the further away this coefficient is from zero, this effect is more dramatic. So we did several runs, and for this specific example, is from the third negative binomial component in the model that has three count components. And what we see here, for example, is that features such as the distance to the origin of replication and the AT content of the gene has a small negative effect towards the mean of this specific negative binomial component. And this goes back to the very good online question about like what does different components mean biologically. So it varies again from data to data, but it is also worth noting that in other negative binomial components, we can see that the same gene features can be affecting the means in different ways. So is it because it's through a different pathway, through a different mechanism, or this is a totally different subpopulation? That is like a question that is interpretation that we need more investigate on. But it is interesting definitely to see that there's a level of data complexity here that is accounted for by having more than just one count component. So this is the first way an independent variable can be fitted to a mean and therefore affect the counts in the end.
It's Manisow.
It also fixed my screen, so that works out nicely in a way. So we're just saying that there are two ways. And the second way an independent variable can affect the count outcome is through the component probability from which a data point is generated. So in our mock example, that would be, well, that was loud, sorry. That would be the probability distribution of like which cup you end up grabbing. And in this case, if you, for example, have an independent variable that increase your probability of getting the non-functional cup and give you a zero count, then your spacer count is gene expression, which is recount per gene here, is expected to be lower. So with this second method where the independent variable can affect the gene expression counts, we can fit these variables to the component assignment probabilities. And again, if we look at the value of coefficients, and this time we have also the zero-inflated component here. One thing that is really interesting to me at least is where the anaerobic respiration function membership has quite a high negative coefficient, which means that it has like a strong negative effect on the counts. And that means if a gene is annotated with an anaerobic function, it is less likely to end up being a zero count. In our data, it makes sense because it comes from a biofilm anaerobic culture. So expressing those genes is certainly helpful in that environment. So this is the second way where a gene feature can affect our outcome. And it's really interesting, especially looking at personally find it really interesting to look at the zero-inflated component probability coefficients, because that really like clues you in what are the features that probably has the biggest effect at the very fundamental level of like, do you end up with a zero or like a count? And that is a very interesting part for me to check out every time I do a fit. Jeremy?
Rucheng, I'm sorry, you may have covered this. How are you dealing with the potential issues of identifiability that could seem to me clearly arise here?
Mhm.
Like if negative binomial component 3 shifts an estimate downward, 2 could just shift it upward.
Yeah, so So in our model, most of the time we use a soft assignment, which means that, like you said, if negative binomial component 3 has a high mean, and we multiply it with the probability that the count comes from it, and which can be counteracted with like a lower mean from a negative binomial component, like 2, for example. So one thing is that they are all positive counts, so good thing that we don't get a completely like negative effect. And also with a soft assignment, we end up doing that because it kind of makes sense numerically. We also have results that is from the hard assignment that has similar results. The hard assignment is that you just only assign it to the component with the highest probability. And in some of the data we are currently working on, hard assignment is having a difficult time because of the dispersion of a negative binomial component tend to push it to very low counts, for example. So going back to the identifiability that we're talking about, so we can consider each count here is like a composite of all the effects from different negative binomial components. But if we use hard assignment, we can more focus on like a single independent, a single component's effect on the count. Does that help?
Somewhat, I guess I
Yeah.
can see potential strengths to what you're doing here. If there's sufficient evidence in the data to allocate certain identifiable
Yeah.
associations of the data to specific components of your model, that's really interesting.
Yeah.
Just
Yeah.
with the width of these, I'm concerned there's not. You know?
Yeah, so this is FIFO cross validation data.
Yeah.
So you can see some variables, well, some components end up with like a tighter distribution, which means that in every fit, it pretty consistently end up with the same results. And some of them are not. And we're thinking something more like there is a group of gene or the subpopulation that is more consistently like a core gene or core population that has similar statistical properties.
Yeah.
So like I said, some of them have like a wider range of like where the coefficient end up landing on. And I think it is worth doing more like validation type of analysis and see how much we can trust if we use like a hard assignment, for example.
Thanks.
Yeah, nice. All right, so that is basically how a gene feature can contribute to the observed expression levels, either through the means as we covered previously and through the component probability here. And what we have landed on at this point is that we have a model that can connect our independent variables with the outcome. And one thing we are really glad that this statistical model being able to do is that it's more a flexible model. So you can basically connect any features that is connected to account-based data. And specifically, it has some particular advantage to, of course, zero-inflated data and also integers count data that end up with a lot of small integer counts that doesn't seem to fit into a certain classic statistical distribution. So with that, at this point, we started with the data where we want to check out the acquisition preference across the source genome, and we established some chromosomal features that correlated with the spacer counts. And those are the data that led us to the tools that we use for zero-inflated negative binomial model and found that it's insufficient for our purpose. And we developed our own tool for a flexible model for a zero-inflated negative binomial mixture model. So going back to where we started, our next step is definitely apply the modeling framework to where the question all started, the spacer counts and the genomic features. I haven't got all the results back yet, but I'm very excited to see what we can see here. So that part is the statistical modeling part. And to sidestep a little bit, at the beginning of this presentation, Marcin and I were talking about how nice it is to be able to present a tools that is still under development, that is actively still being worked on. And I want to talk about some implementation side of things of this modeling framework that still have room for improvements. So as you have seen, the history of this model started with a data analysis project and end up landing on an optimization problem. So given this history, it was written in R. And we soon ran into a couple different challenges. For example, we end up having to use, because of the model itself, derivative-free optimization, and we need tools for that. And we also observed that our optimization landscape is a lot more jagged than the optimization problem the algorithm that R comes with can handle. So that's when we asked if we can find a different optimizer that performs better than what we have. So we landed on never grade, which is never grade says the grade stands for gradient. It doesn't mean that you never graduate. But it's actually written in Python, and it's an open source tool by the Facebook research team. So in this convoluted history, we end up having R into never grade and go back to R. So there are a couple of disadvantages, of course, in this process. So the first thing is that we unnecessarily have to transition between coding languages. And that also means the environment is very hard to maintain. You basically have to maintain a good Python environment and a good R environment and hope that they work together. And the runtime is not ideal. Neither Python or R is known for, fast runtime. And especially in R, it's very hard to manage computation resources. So what we are doing right now is that we're switching to NLOpt for a control random search algorithm instead of never grade. We have some benchmarking results that shows a control random search algorithm in the majority of the cases perform better or as similar as never grade. And it has a lot more option when it comes to which programming language we are going to use, because it is implemented with a lot of interfaces from different languages. And from there, we are refactoring the code in Rust. So hopefully it will help us to get a better standalone tool that is good for release and also easy to install and use. And we certainly want to take advantage of the flexibility of the model and eventually look into more different types of data. For example, we can imagine the flexibility of the model would do well in ecology or metagenomic data, for example. It will be interesting to see how this developed tool can work under those situations. So once in a while, it's fun to talk about implementation of the tools. So that is my presentation. I want to acknowledge especially everyone in Fredolino Lab for all the help, and also DCMB and the Department of Biological Chemistry, and also funding that makes the research possible. And with that, that's all that I have for today, and I will take any questions.
If anyone online has a question, you can put it in the Q&A box.
I guess I have a question about kind of the initial problem that this started from. So
Yeah.
it looks like you made a great model for that. You've got the zeros and then the slope coming down.
Mhm.
But in the genomic model, you've got, it looked like 3 really big peaks. How can the zero-inflated model you made kind of come back to that? It looks so different from, my view at least.
Yeah, so let me think about it. So in the gene expression data example, you're mentioning that it looks like there are 3 bigger components in each time. So that is when we look at the effects separately. If you kind of like call back to the original count distribution we see a lot of 0, but it roughly look like a negative binomial distribution, which is the tricky part. So a mixture model doesn't necessarily end up with, like, for example, a 3-component that you see 3 peaks. Sometimes they look like something that is just from 1 component. We test components from 1 to 4, and we end up selecting 3 as appropriate for the data because the likelihood of the model improves, basically says the model fits better to the data, but at the same time, it's not overfitting because we check the information criteria. When we reach 4 components, it starts to overfit. The extreme cases that you fit each data point with 1 model. So that's how we end up landing on 3 components. So that's also one thing that's kind of amazing to me because visually it really looked like just a negative binomial distribution. But when you actually fit data and look at the number, it has complexity that I cannot see visually. So when it comes to spacer count data, I think what you're thinking is the distribution of it across the genome. But if we plot the count itself by the number of genomic sites, we'll end up with a histogram that probably look like something that we'll see in the count component or something totally different. That's why like a good flexible model can help with this process to figure out what actually works. Yeah,
That makes sense. Thank you.
of course, thank you.
Does this model for spacer count, is there any possibility that it varies by the genomic location that we're looking at?
Yeah. So that's basically the biggest question we are asking is that, well, we have a spacer count associated with 1 genomic location. We want to see if any feature associated with that genomic location affects the spacer count. For example, if it has like higher AT content near that location, or if it is landed in the gene or not. And those can become part of the gene features that goes into the regression model. So at the end of the day, we're basically building a model that explains the spacer counts by those like genome position dependent features. So basically what you're asking is the biggest question we're asking, that when we eventually refactor the code and start to apply the data to apply the model to our spacer data. So that will let us know not just by the coordinate of the genome, but by the feature around a genomic site. So I hope that makes sense.
Yeah.
Thank you. Yeah,
So Rucheng, you mentioned I think you call it soft selection,
soft assignment, yeah.
soft assignment. Are those assignments global or do they vary by genome position?
They vary by genome position because you can also, so this model, if you want it to be global, you can fit it with only an intercept and no independent variables that varies across different genomic locations. Then you get something that is global. But if you fit independent variable to the assignments, because for example, the measurement of AT content varies from position to position, and in your model, this assignment will end up varying from position to position. So the flexibility of it, I guess it comes down to. And you can either fit just the intercept, then everyone gets like a 20%, 50%, 30% distribution, for example. But then if this distribution is fitted with the independent variable, then each independent data point will end up with a different predicted component probability.
Yeah, so I guess that makes me really curious. Have you looked at how those probabilities change over the genome? Like, are there specific components that predominate in the peaks? And
So I haven't specifically looked into that, but I think that is part of what we're trying to get at when we show the result with the zero-inflated component assignment probability, is that for a zero-inflated component, it has like a more easily interpreted kind of aspect to it. And with different count components, it's definitely worth looking at like we're actually for the same data, we have the go term analysis of each one of them assigned to different components. We are redoing that analysis because we want to do it more like a differentially expressed way by fitting the model, considering the biological condition it was done in for the experiments. But we can definitely do like by component to component analysis, if that's what you're thinking.
I think I was just thinking more
Yes.
simplistic than that, just, you know, is there something,
Mhm.
does component membership,
Yeah,
is that predictive of whether you find yourself in a valley or in a
Mhm.
peak? in
oh, I
your-
see what you mean, yeah. I think given the means vary from different components, if you have a higher probability, have a membership with a component with lower mean, you end up more likely to be in the valley. So I think this definitely related, but numerically, how it is related, I haven't looked into it yet.
It's cool that it's like a interpretable dimension reduction.
Yeah, that would be fun, yeah.
Cool.
Any other questions? Got a comment from Maureen saying very cool work and thanking you.
Thank you.
I'm not seeing any other questions online or in the room, so let's thank.
And thank you all for the questions. It's always fun to present with feedbacks and just hear everyone's thoughts, so.
Thanks everyone for coming and I hope to see everyone next week.
I'm really